Imagine a world where the very air you breathe is brewing the building blocks of life. That's exactly what scientists are now suggesting about early Earth. A revolutionary new study challenges a core belief about how life began, suggesting that the primordial sky was far more involved than we ever thought possible. For years, the prevailing theory was that complex sulfur-based molecules, vital for life, only appeared after life was already established. But here's where it gets controversial. Researchers at CU Boulder, along with their collaborators, published a groundbreaking report in the Proceedings of the National Academy of Sciences suggesting something radically different, that Earth's early atmosphere was a veritable factory, churning out these crucial molecules billions of years ago, before life took hold. According to the study, the young Earth's atmosphere, bathed in sunlight, was capable of generating sulfur-based molecules. These molecules are now recognized as fundamental components of life as we know it. This discovery effectively flips the script on the traditional understanding of how life's ingredients came to be. Nate Reed, a NASA postdoctoral fellow who conducted the research at CU Boulder, emphasized the significance of the findings, stating, "Our study could help us understand the evolution of life at its earliest stages." This research provides a new perspective on the origin of life and challenges the established scientific narrative. Why is sulfur so important, and why should we care about these findings? Think of sulfur as a sibling to carbon. Both are essential elements present in every living thing, from the tiniest bacteria to the largest whales, and yes, even us humans. Sulfur is a key component in certain amino acids, which are the fundamental building blocks of proteins. These proteins are the workhorses of our cells, carrying out countless vital functions. The traditional view held that while sulfur was present in the early atmosphere, it was only after life emerged and organisms began producing organic sulfur molecules, like amino acids, that these molecules became readily available. Earlier attempts to recreate early Earth conditions in the lab often fell short. They simply couldn't generate significant amounts of sulfur biomolecules before the simulated emergence of life. And when they did appear, it was often under highly specific and somewhat unrealistic conditions. These conditions wouldn't have been commonplace across the planet. And this is the part most people miss, the idea of needing widespread availability. The scientific community's excitement was palpable when the James Webb Space Telescope detected dimethyl sulfide, DMS, a sulfur compound produced by marine algae, in the atmosphere of exoplanet K2-18b. Many scientists considered this a potential sign of life beyond Earth. However, previous research led by Reed and Ellie Brown, a chemistry professor at CU Boulder, showed that DMS could form naturally in the lab, using only light and simple atmospheric gases. This indicated that DMS could exist on planets without life. In their latest experiment, Brown, Reed, and their team decided to investigate the true potential of early Earth's atmosphere. They meticulously recreated pre-life atmospheric conditions by illuminating a mixture of methane, carbon dioxide, hydrogen sulfide, and nitrogen. Working with sulfur isn't easy. As Brown pointed out, sulfur tends to stick to laboratory equipment, and sulfur-based molecules exist in incredibly small concentrations compared to common gases like CO2 and nitrogen. You have to have equipment that can measure incredibly tiny quantities of the products, she explained. Using an incredibly sensitive mass spectrometer, the researchers were able to identify and measure the chemical compounds produced in their early Earth simulation. The results were astonishing. The simulation yielded a wide range of sulfur biomolecules, including the amino acids cysteine and taurine, and coenzyme M, which is crucial for metabolism. The research team then extrapolated their findings to estimate the potential cysteine production of the entire ancient atmosphere. Their calculations suggested that early Earth's sky could have generated enough cysteine to support approximately 1 octillion, 1 followed by 27 zeros, cells. To put that in perspective, modern Earth contains roughly 1 nonillion, 1 followed by 30 zeros, cells. While it's not as many as what's present now, that was still a lot of cysteine in an environment without life. It might be enough for a budding global ecosystem, where life is just getting started, Reed noted. The researchers propose that these atmospheric biomolecules may have rained down onto the surface, essentially delivering the chemical ingredients needed to kickstart life. Life probably required some very specialized conditions to get started, like near volcanoes or hydrothermal vents with complex chemistry, Brown said. We used to think life had to start completely from scratch, but our results suggest some of these more complex molecules were already widespread under non-specialized conditions, which might have made it a little easier for life to get going. This research raises some profound questions. Could life have emerged in multiple locations on early Earth due to this widespread availability of building blocks? Does this increase the likelihood of finding life on other planets with similar atmospheric conditions? And, perhaps most controversially, does this mean the origin of life wasn't as improbable as we once thought? What do you think? Share your thoughts in the comments below.